This year's prize deals with cells and sensibility. The Royal Swedish Academy of Sciences has decided to award the 2012 Nobel Prize in Chemistry to Professor Robert J. Lefkowitz at Howard Hughes Medical Institute and Duke University Medical Center, Durham, North Carolina, USA, and Professor Brian K. Kobilka at Stanford University School of Medicine, Stanford, California, USA. And the Academy citation runs for studies of G-protein coupled receptors. Professor Sven Ledin will now give us a short summary in English. Please, Sven.
Thank you, Stefan. Boo! Do you remember the last time you got really scared? The dryness of the mouth, the heart that skips a beat. These are signs that your body is getting ready for flight or fight. Adrenaline surges through the system, and it affects metabolism, circulation, respiration, muscle tonus, and vision. It leads to an orchestrated response from the billions of individual cells that make up our bodies. It was known for a long time that adrenaline does not enter into the cells it affects, but an increase in the adrenaline levels on the outside of the cell leads to a response at the inside. A receptive substance, a receptor, was correctly assumed to be involved, but the nature of this receptor and how it acted remained a mystery for a long time. Now, thanks to the work of Robert Lefkowitz and Brian Kobilka, awarded this year's Nobel Prize in Chemistry, we know what this receptor looks like in the finest molecular detail. We also know that it's just one member of a huge family of receptors, the G-protein coupled receptors, or the GPCRs. We know the mechanism by which GPCRs function, and we know how that function is regulated. Now, the communication between our cells is essential not only in times of fear, but in everyday life. And unbalance in this communication leads to unhealth. Now, a large proportion, some say 50% of all pharmaceuticals used today, rely on action targeting GPCRs. So knowing what they look like and how they function will provide us with the tools that can help us to make better drugs with fewer side effects.
Thank you for these words, Professor Ledin. And now, Professor Snugrep-Lindsey, you will give a more detailed presentation of this year's prize.
Thank you. G-protein coupled receptors sit in the membrane. They tell the inside of the cell what's going on the outside. Thanks to the studies by Robert Lefkowitz and Brian Kobilka, we know how these receptors are built, how they work, and how they are regulated. There's a whole family of receptors that are built in a very similar way. They all have 7 helices, spiral-like structures, here depicted as rods that go through the membrane. Therefore, they are also called 7 transmembrane receptors, 7TM. In our body circulates a number of neurotransmitters and hormones. Some of the names you may recognize: serotonin, histamine, adrenaline, etc. And these action via G-protein coupled receptors. The molecule to the left, adrenaline, binds to at least 9 different G-protein coupled receptors in our bodies and cause different responses in different organs. So when the heart and lungs muscles are activated at the same time, our digestion is shut down. The molecule to the right is man-made. It's a beta blocker. It's similar to adrenaline, yet different enough that it only binds to a subset of the adrenaline receptors. Therefore, it only affects certain organs. Indeed, the G protein-coupled receptors are the targets for about half of all pharmaceutical drugs made today. And these are used in treatment of conditions like high blood pressure, neuropsychiatric disorders, Parkinson's disease, migraine, gastric disorders, you name it. Think of a disease, and there is probably a medicine there affecting a G protein-coupled receptor. So the receptor sits in the membrane, and when a signal comes here in the form of increased adrenaline concentration, the task for this receptor is now to tell the inside of the cell that this has happened. But it doesn't do so by letting adrenaline in. You can see no adrenaline inside the cell. Instead, the receptor binds, each receptor binds one molecule of adrenaline in a pocket on the outside region. And this leads to a change in the shape of the receptor so that it opens up a site in the inside where it can bind another protein, for example, a G protein. Here it's depicted in red with its alpha, beta, and gamma subunits. The receptor is a little bit like a bundle of rods. When the receptor is not activated, the rods are fairly parallel to one another. And on the inside of the cell, there's really nothing to interact with. When a small molecule binds at the outside, rather small changes in the structure here propagate into a much larger change on the inside, where the G protein can bind. This is also the reason another small molecule can elicit very different changes on the inside, which then signals through other proteins on the inside and other signaling pathways. The complex we see here is the active signaling unit. It is sometimes called the ternary complex because three things are needed to come together: the hormone, the receptor, and the G protein. This has been known since the middle of the 1980s, thanks to studies using, for example, radioactive hormones, cloning work. Today, we know what this complex looks like in near atomic detail. What you see here is a molecular masterpiece. It's the result of 3 decades of dedicated biochemical work at the bench. The methods derived to be able to obtain this structure by crystallography have now also been used and are being currently used to make crystals of a large number of drug-important receptors to derive their structures. The receptor in the membrane is something like a switchboard in the wall of a building. The operator of the switchboard speaks more than one language. Therefore, clients of different nationality can call the same switchboard, but the operator is experienced enough to send a signal in different directions depending on who calls. This is exactly like a G protein-coupled receptor. Depending on what small molecule binds to the outside, the signal is sent in different directions inside the cell. This is very important knowledge in development of drugs to obtain medications with fewer side effects. Each operator speaks only a limited set of languages. Therefore, a number of switchboards are needed to serve all the clients in the world. We have about 1,000 different G protein-coupled receptors in our body to be able to sense a huge number of different hormones, neurotransmitters, and other signals. Some clients can call more than one switchboard. In our body, adrenaline can interact with 9 different receptors and cause very different responses in different locations. G protein-coupled receptors are also found on the outside of our body, in the sensory cells, in our nose, on our tongue, in our eyes. And this allows us to sense our environment, to smell, to taste, and see. Yeah, now I really need a cup of coffee. Thank you. There you are. Thank you. Thanks to the G protein coupled receptors, I can now really enjoy this cup of coffee. The smell, the aroma, the beautiful serving, neurotransmitters, hormones, many G protein coupled receptors are active now. And thanks to the studies by Robert Lefkowitz and Brian Kobilka, I can also enjoy the excitement of knowing exactly, in finest molecular detail, what's going on in my body right now when many signals are passed over my cell membranes. Thank you.